Hello, we're working today with changes in DNA. Thank you for C-Scope for providing PowerPoint slides and images. This is going to be a probably hard to understand video audio-wise because of my computer fan. So if it's too distracting, this evening I will be redoing the post so that you can hear me better with a microphone. So you can either wait until that happens or go ahead and muscle through tonight and get your homework done. All right. So the first thing that we need to talk about is an analogy to help us remember and understand some things about DNA. The sentence I want to talk about is the fat cat ate the wee rat. So just in case you're not sure, wee, W-E-E, means small or little. Okay? This sentence represents a strand of DNA. So if we were to pretend this is DNA, and the word is each codon. Okay? And that is why they're in groups of three, or they're three-letter words, or why we chose those letters or words, is because each strand of DNA has codons, and each codon has 3 nucleotide bases. So if the words are our codons, then each letter is a nucleotide base or a nitrogen base. Okay? Now mutations. Mutations are things that we think of as maybe a change. We think of them as mistakes. They have a negative connotation, but they are not always negative. You may think about Teenage Mutant Ninja Turtles, you may think of the X-Men, or growing a third eye, or having an extra arm, but it's usually not as dramatic or as awesome as that. It just means a little bit of change, and in this case, your DNA. Now, what we're going to know, knowing what a mutation is one thing. Knowing that a mutation has different types, and knowing what those types of mutations are is another. So the first type of mutation that we're going to talk to is substitution. When you think of substitution, you think of maybe a substitute teacher who comes in and replaces your teacher for a small amount of time, and that will either be fine, just as good as your teacher, or maybe not so good. So let's look at this. The fat cat ate the wee rat was our original DNA. If we have a substitution mutation, an example of that might be the fat cat ate the wet rat. Now, it's still a codon, it's still a sentence, it still makes sense, but now instead we're saying a little rat, we're saying there's a wet rat. All right? So the E here, this E base changed to a T, but our sentence still makes sense. So we have, this represents the codon is being changed, and that the nucleotide base is being changed. And this is going to affect the protein. Either it will code for a different amino acid here, or it could code for the same thing. So, and we might not see a change. So maybe it's okay, maybe it's not. A deletion mutation is just like the delete button on your computer keyboard, and you delete a nucleotide base, it shifts things, right? So if we have the fat cat ate the wee rat, now we have the fat cat at at. Now, if you're not paying attention to the screen and you're just listening to me, you're like, what language is she speaking? Exactly. If you see here, we've eliminated this E. Well, the rest of the whole sentence shifts down, and it changes what's going on here. It's changing the meanings of these words. Okay? And how would this change? Well, the fat cat, those amino acids are going to be fine, but starting here, ATE being changed to ATT is going to probably give us a different codon, and HEU is probably going to give us a different amino acid than the, and ear may give us a different amino acid than we, and at may not give us amino acid at all because it's missing its third nucleotide base. So this is going to either break down the protein earlier than it should, or it may code for the wrong type of protein. right, insertion. This comes from the word insert, or to put in between. So here we have the fat cat ate the wee rat, our original DNA strand, and now we are changing that to the fab to ka tat fu era t. Well, that doesn't make very much sense at all. So when we insert a base, like here, we have the T is now replaced with a B, and we push all the rest of the bases down, we've inserted this B. Well, fab makes sense, that's a word, but it changes all the rest of the codons so that they don't make sense. And that's going to change your protein, so it does the same thing. It'll either end the protein early, or it will not function in the way that it's supposed to. All right. Now, there's two different types of mutations, or the mutations can be put into different categories. Point mutation and frame shift mutations. A substitution is a point mutation because it only affected one nucleotide and not the rest of them, and so it was lesser of a big deal. And the last two examples, deletion and insertion, where it made the rest of the amino acid chain incorrect or the DNA not make sense, those are considered frame shift mutations. So if you only affect one thing, that's called a point mutation, and if you affect more than one codon, then it's a frame shift mutation. So, what is the difference between a point mutation and a frame shift mutation? How much of an effect it has. Obviously, looking at the next question, which one has the most significant impact? That's going to be your deletion and your insertion, because they're frame shift and they're actually changing the language that we're trying to code proteins in. There's one other thing that's a point mutation, and it's called a synonymous or silent mutation. It does not change or cause a change in the amino acid sequence. If you remember back when we were coding for proteins, there was sometimes more than one codon that would code for valine, an amino acid. So if we make the mistake and we still get valine, is it no harm, no foul? Are we okay? So it can change it enough that it disrupts the functioning in the body, but it's not as much. So for example, TAT changed to TAC, they still coded for tyrosine, and then CTC also, if we made a mistake and had a point mutation as a CTA, they still code for leucine. So that's what we call silent mutations. They're still mutations, but they happen silently. They're used to say that it's not a big deal if it's a silent mutation, no harm, no foul. Now they're saying that there's a bigger issue here. Yeah, you get the same protein, but not as much of the same protein, and that can lead to issues depending on what the protein is needed for. All right, inversion. Inverted means like inside out. So if you were to wear your shirt inside out, we would call it an inversion. So here we have the fat cat ate the wee rat, and then all of a sudden we look at the fat tar u eta tac. Well, that doesn't sound like English, but as you see, we've inverted. Here's cat, now cat is backwards and over here. So it's the same number of nucleotides, which we didn't see in a frame shift mutation where the whole thing shifted over more or shifted one less, but we are seeing that it still doesn't make very much sense because we've inverted the amino acid sequence. This would affect the protein. Pretty big deal, because tar is not going to give us the same amino acid as cat. And so you may have some that code for the same amino acids, but it would be rare. All right. Looking outside of the DNA, we have chromosomes. Remember, a chromosome is kind of like a dreadlock of DNA. So we still have DNA going on in here, but it's on a bigger scale. So instead of making a mistake on a quiz, now we're making a mistake on a test. It's a bigger issue going on. As you can see in this mutation, what has changed? I see that it's gotten longer. And not only has it gotten longer, but if you look at the band pattern, that's the same here and here. So we've copied this gene or these segments twice. what could happen here? Our protein's going to end up being a different size. All right. Chromosomal mutation here, we can delete as well. If we X this, this is going to become shorter, and whoever gets this chromosome is not going to be able to carry out those instructions, and so that can lead to issues as well. All right. The next one is translocation. This is where you shift parts of a gene to a different part of the gene, and then there's issues. If you kind of think about Easter eggs, if you have a solid yellow Easter egg and a solid blue Easter egg, and then you switch the top and bottoms of them, then you have this hybrid Easter egg. Now, in that case, the Easter egg works just as fine. In this case, it may or may not, because it depends on what these code for and how important they are. This is called a translocation, where we move one segment of a chromosome to another segment of a chromosome. Okay. Non-disjunction. This is, the example I'm going to go over is called trisomy 21, or Down syndrome. This picture that you're seeing here is called a karyotype. Okay? And with a karyotype, this is all your chromosomes. We have 23 pairs, or 46 total. So like if you have 23 pairs of shoes, you actually have 46 shoes. You're supposed to have a pair for each chromosome number. So if you look at this first, they're all paired up beautifully, and so they're perfect. Same with the second row. And then let's talk about the last row. Here you see that there is 3. Well, just like it doesn't make sense to have 3 of the same shoe or 3 of the same sock, this is not paired correctly, so there is a mutation called non-disjunction. The other part of this, if we were missing one of these, just like it doesn't do you any good to have only one sock or one shoe, then that's not going to work as well. Over here you might see this. Boys and girls are different in many ways, and on the chromosomal level, girls have XX chromosomes and guys have an XY chromosome. So this Y is what makes them a guy, and that's kind of the cheesy way that I remember it. Y, you have to have a Y to be a guy. Otherwise, the ladies are just stuck with these X's here. All right. This comes from an error in meiosis. So somebody's, whether it was the male or female parent, had an extra chromosome 21, and they gave that to their baby, plus the other parent had the normal amount of chromosome 21. He gave 1, and maybe she gave 2, and so if there's 3, then the child would have Down syndrome. Okay. A mutation impact. The impact of the mutation is on an individual, depends where and when it occurs. If there's a mutation in the DNA of a zygote is a baby. And so how would that impact the individual? If it's on the chromosomal level, it can be a huge issue. If you delete the wrong chromosome, the baby will not survive the pregnancy. And sometimes that they can survive the pregnancy until they're born, and then they can't last outside of the mother. So this can be an issue. If there's a mutation in your skin cell, that can affect what proteins are made for the skin cell. It could unregulate the cell cycle and maybe cause cancer. But if your skin cells are not making the appropriate proteins, then they're not going to be able to repair and heal themselves as much. All right. The last thing that we need to talk about is a pedigree chart. A pedigree chart is like a genetic family tree. They're not as lovey-dovey as your family tree, but you can track conditions based on a family history as much as you can doing gel electrophoresis and karyotyping. So we use family information to trace genetic conditions that have been passed along from the family, and genetic conditions are caused by the mutation. The genetic counselors use them to help people analyze their family history and determine the probability of having or passing on a particular condition or mutation. So if you work with a genetic counselor, you and your mate would be able to see if there's a likelihood based on family history of certain genetic conditions. All right, so ways to read this. If it's an empty square, that's a guy, a male. All right, so guys are square. And if it's a circle, that is a female. If it's a darkened square, that person is affected by the mutation. If it's a darkened circle, then that is a female that is affected by the genetic mutation. And if it has a slash through the circle or square, that person is deceased or dead. All right. And so you don't have that person to physically ask if they have that condition or test them. And maybe it's only based on, yeah, my grandpa told me that my great-grandpa had this condition. And so it's not as reliable. An example here, we have a red square. Here's our guys that are squares and our girls that are female. We have a key where if it's affected, they have red squares and circles. The non-affected are blue. So we can see that this couple, this guy and this gal, they had one boy who was affected, one boy who is not affected, a girl who is affected, another girl who is affected, and another girl who is not affected. Okay? And then they each have their own children. If the circle does not connect to the original parents, that is the person that mated or married in with this particular offspring. Okay? So all the people that married in, this wife was not affected. This wife was also not affected. This husband was not affected as well. All right. If they don't have a partner, then that means at the time of the karyotype they had no children. And it makes sense. The gene was passed down to a boy and two girls. This family that grew, the grandchild, children did not have anybody affected. This family, one girl was affected and the boy was not. And in this family with 4 children, a boy and a girl were affected and a boy and a girl were not affected. So what we can see from this is that it's not sex-linked, because if it was a sex-linked gene, then if the guy had it, then we would only expect to see boys having it. If the girl was affected, then we'd only expect to see girls having it. So we consider it's an autosomal mutation that's happening, which means it can have from male to female. And the other thing that we can see is that it is not a recessive trait. It's probably a dominant trait to have 1, 2, and 3 people get the trait. So whatever this affected condition is, genetic mutation, then it's a dominant gene. And that concludes our lesson for today. Again, I will be reposting this, hopefully with a better recording this evening, and I hope you do well on your quiz tomorrow. Bye-bye.